Good morning, dear children. Today we are going to study fertilization in angiosperms. Fertilization, it is the fusion of male and female gametes. When we are going to study this fertilization in angiosperms, first of all we have to see the embryo sac and the different nuclei associated with it and the fusion of the gametes. And here we can see in the case of embryo sac or female gametophyte, when it is in the 8-nucleated condition, here two centrally placed nuclei are there called polar nuclei. Then one egg apparatus is egg and synergids and the 3 antipodals. And at the time of fertilization, we can see the polar nuclei fuse together and form secondary fusion nucleus. And here the egg fuses with one of the male gamete. And here you can see in the case of angiosperms, the fertilization is known as double fertilization or triple fusion. It is called double fertilization because from a single pollen grain, two male gametes are produced. One of the male gamete, it fuses with the egg and form the zygote. One of the male gamete fuses with the egg and form the zygote which develop into embryo. And the second male gamete, it fuses with the secondary fusion nucleus. That is, the polar nuclei fuses together form secondary fusion nucleus, and that the second male gamete fuses with the secondary fusion nucleus formed by the fusion of the polar nuclei and form what is called the secondary fusion nucleus and form what is called the primary endosperm nucleus. Thus we can see two gametic fusions are there and also three successive fusions are also there. Since there is two gametic fusion, it is called double fertilization. That is the first fusion of egg with the male gamete resulting in zygote. The second is the fusion of the secondary fusion nucleus with the male gamete. And there are two gametic fusions and three successive nuclear fusions, that is egg and male gamete 1. Then second fusion of two polar nuclei resulting in secondary fusion nucleus. And the third fusion is the secondary fusion nucleus plus the male gamete, second male gamete resulting in primary endosperm. And this primary endosperm nucleus later on develop into the triploid endosperm proper. And double fertilization or triple fusion is a unique feature of flowering plants or angiosperms. Next we have to study development of dicot embryo and the structure of dicot embryo. The embryo develops from the zygote formed by the fusion of male and female gamete. And this single cell now undergoes an unequal division resulting in two cells, one basal cell and a terminal cell. The basal cell by repeated division develops into the suspenser for absorbing food materials, and the terminal cell first of all develop into a globular embryo, this like this, and then into a heart-shaped embryo and embryo proper with two cotyledons and embryonal axis with a plumule and radicle. Plumule and radicle. Thus we can see the zygote formed by the fusion of male and female gamete, which is diploid in nature. First of all divides into two cells and one basal cell and a terminal cell, and the basal cell by repeated division develops into the suspenser and the terminal cell develops into First of all, the globular embryo, then into heart-shaped embryo and embryo proper with two cotyledons and embryonal axis like this, and this suspensor dries up. And now we can see when the embryo is fully mature, it is provided with two cotyledons and the embryonal axis with the plumule and radicle. The plumule later on develops into the shoot system and the radicle develops into shoot system. And here the radicle is protected by a root cap also. And this embryo can also be drawn like this. Here two cotyledons. This is the plumule. Here it is the plumule. And here it is the radicle. We can draw the embryo like this also. Two cotyledons, plumule and radicle. You know, the embryonal axis just above the cotyledon is known by the name epicotyl. That is the portion of the embryonal axis just above the cotyledon. This is the cotyledon, and you can also put just above that region. This region is called epicotyl. And the cylindrical region just below the cotyledon is called hypocotyl. Epicotyl and hypocotyl. Thus we can see in a dicot embryo, two cotyledons which store food materials and become thick and fleshy, and the embryonal axis having plumule and radicle. The plumule develop into the shoot system and radicle develop into a root system. And in the condition we can see the plumule is protected by the root cap. And we can see the portion of the embryonal axis just above the cotyledon is called epicotyl, and just below the cotyledon is called hypocotyl. This much about the dicot embryo and its development. Next we have to study the structure of monocot embryo. You know, in the case of monocot seed, the major portion of the seed is formed of endosperm, and the embryo is very small and seen towards one side of the seed. And here you know, this is the structure of monocot embryo. There is only one cotyledon which is known by the name scutellum. In the case of monocot, there is only one cotyledon, and the cotyledon is known by the name scutellum. And the embryonal axis consists of the plumule and radicle. And the embryonal axis contains some leaves, primordial leaves. And here we can see this plumule is covered by a protective covering called coleoptile, and the root portion or the radicle is covered by a root cap, and also the protective covering called coleorhiza. Here the embryo is very small. Major portion of the seed is occupied by endosperm, and here only one cotyledon is there which is known by the name scutellum, and the embryonal axis consists of plumule and radicle. The plumule is protected by a protective covering known by the name coleoptile, and the radicle is protected by coleorhiza. This much about the structure of monocot embryo. Next we have to study endosperm. We have already studied that the primary endosperm nucleus by repeated division develops into the nutritive tissue called endosperm. There are three different types of endosperm, namely free nuclear type, cellular type and heliobale type, of which the free nuclear type is the most commonly seen endosperm. Here we can see First of all, the endosperm nucleus divided by repeated nuclear division into a multinucleate structure, and then wall formation occur from the periphery and develop into endosperm. Thus, the three types of endosperm development are free nuclear type, cellular type, and helobial type. And this is the endosperm, which is the nutritive tissue for the developing embryo. And here, in the case of free nuclear type, the single nucleus present in the endosperm undergoes repeated division to form a large number of free nuclei, that is followed by wall formation and develop into the embryo. Here, in the case of cellular type, whenever the nucleus is dividing, that is followed by wall formation. And helobial type is an intermediate type of embryo endosperm development between that free nuclear and cellular type. It is intermediate type, helobial type. Thus, the three types of endosperm are the free nuclear type, cellular type, and helobial type. Today we can stop here.